Wilson International Center for Scholars on this hot, muggy, oppressive day in Washington in the summer. It's good to see everybody, actually. It's a fairly large turnout. What we're going to do today is release the results of a workshop that was held here in November, actually. It was sponsored by the Department of Energy and the Alfred P. Sloan Foundation. And Lori and I are going to take you through the report, and then we're going to kind of open it up to questions. We were the co-chairs for the workshop process, and I think Lori took probably the most notes, so she's going to start. She's with the Health Law Institute at the University of Alberta, and also is a private biotechnology policy, law, and ethics consultant. So I'll have Lori sort of go through the report, give you a sense of how the workshop was run, and kind of what started to come out on the back end. And then I'll take you through sort of the gap analysis that we tried to do in a day and a half, and then talk a little bit about what we're planning to do and just in terms of follow-up, because there's a next step and probably a next step to this that will keep going. So let me turn it over to you.
Thanks, Dave. Thank you all for coming. We really had a very good time in the 2 days that we were together for this workshop, and like many workshops that are aimed at thinking about the future and anticipating, we ended up raising many more questions and developing some really great ideas for research agenda in the area of synthetic biology than providing really concrete answers, which is the way I think it's supposed to be. So we have, as Dave already mentioned, the workshop was organized by the Department of Energy and the Alfred P. Sloan Foundation and hosted by the Woodrow Wilson Center. Actually, in this room we met. And we brought together actively practicing synthetic biologists as well as a lot of other interdisciplinary scholars, and all of them were actually working in synthetic biology in one way or another, many of them on the societal issues, which is something that is actually actively brought out in the report, which I'll talk about in a minute. And the idea was to try and really look farther down the horizon than the immediate things that we hear about and that are being published, and try and anticipate with our imaginations on what in fact could happen, and if particular applications came to bear, what might the societal implications be, how we could think about those things, and what kind of structures might need to be in place to be able to aid public engagement in a meaningful way. So the first thing I'm going to do is walk you through the actual structure and methodology of the workshop, and then I'll backtrack and flesh out the various pieces and bring to you some of the substance that we came across in each of those pieces. Prior to the invitees coming to the workshop, we distributed to them a paper that we wrote on systemic risks and other disruptive technologies and implications that those have raised for governance. So there was kind of a background thought piece to get the workshop attendees to start thinking about synthetic biology with that background piece. And we also asked them to provide for us one-page written responses to the following three questions. Are there lessons learned from the introduction of past technologies that might be relevant to synthetic biology? So working from analogy, things that have gone before, are there any lessons that we can learn given the introduction of similar or other types of biotechnologies? Have each of the individuals experienced or had conversations with people about what they're doing that have presented them with challenges arising from synthetic biology and society? So had they had any personal encounters or experiences that they thought were relevant in the context of the workshop? And what urgent ELC, that's ethical, legal, social implication, research issues, needs arise from synthetic biology? What ELC research could they suggest that they thought needed to be done in this context as well? And those one-page papers were then sent to all the participants to really start people thinking, that this is, we want people to actually think about their own experiences and to bring that information to bear. We then had plenary presentations, and the plenary presentations were given by two synthetic biologists actually telling us what they were doing and why they were doing it, which was really exciting to listen to. And then another plenary presentation looking at ELC research from a historical perspective, from its introduction and through the Human Genome Project, and kind of the things that had worked and things that hadn't worked as well. And these plenary presentations are actually there's small summaries of them in the report. I don't know if they're available online. Are they? The plenaries?
I don't
The
know,
slides?
we can make them available if you want to do that.
And then one of the very interesting parts of the workshop was that many of the people around the table, probably the preponderance of people around the table, were grantees of the DOE or of the Alfred P. Sloan Foundation, which has been really active in the area of synthetic biology as well. And so unlike what normally happens in a lot of social science research and in science and social scientists research working in the same area, we were able to actually hear from the PIs from all the research projects what they were doing. And that was, that really seems, that's a no-brainer really. We should know what's happening, what everybody else is doing, but surprisingly that was really very novel to actually hear from people on the other side of the country who are working in the laboratory, what they're doing, and the ELC issues that they're actually dealing with actively, and what some completely disparate groups were working on as well. So that was really interesting, and all of those, the synopsises of what they presented are in the report as well, and gives you a kind of better sense of the breadth of research that's actually taken place in the synthetic biology and societal impacts sphere. And I suggest that you might find that interesting reading. We certainly found it interesting to listen to. And then we had focused breakout groups. And the first was to mix up all the disciplines. We created 3 groups, and we asked them to think about the likely first uses. So this is on a shorter time frame, the first uses of synthetic biology and the attendant societal impacts that or issues that they thought that those might raise. And I'll run through some of the findings from the breakout groups in a minute. And then the next day we asked them to divide along professional lines, and we had sort of a legal and economic group, legal regulatory group. We had a public policy group and a communication public engagement group, and those groups were asked to explore and raise the issues that could be expected to arise from synthetic biology research and its applications, and we'll talk about those in a minute as well. And then we finished the day with an exercise where across these boards behind you, we divided these boards into various areas, and we asked everybody to take Post-its and to put up, to fill and help us with what we call a gap analysis, which is to help us find the knowledge gaps that we obviously couldn't cover in a day and a half that could contribute to a research agenda in the future in this area. So the breakout session 1, just to give you a synopsis, we asked this question: what are the potential uses and applications of synthetic biology? And this was really, we had said to people, this is a blue sky sort of exercise. Think broadly, not just the things that you've read about or the things that you know might be coming in 3 or 4 years. And interestingly, across all the groups, the one really common thread was that synthetic biology has the potential to open up to us a tremendous amount of knowledge about biological processes, biological regulation, understanding in a way that we haven't formally been able to understand how biology works across every aspect of biology. That was sort of a very baseline conclusion of all of the breakout groups, that it was really as a platform for future knowledge about how biology works. And of course then that knowledge could be brought to bear on things as disparate as vaccines and environmental remediation, and we had and on and on and on across all the spheres: agriculture, even space and human health. And another theme that came out of that across the breakout groups was that synthetic biology allows for a lot of interesting convergences between other technologies. So convergences with nanotechnology, for example, that might lead to some very interesting personalized medicine applications where you have sort of radically altered cells and you're able to actually tailor new novel cellular mechanisms to work in the human body. And even convergences with things like microelectronics, about which I know very, very little. And so that was a very interesting exercise. And the other thing that that we asked them to do was to look at the potential, what we called game changers, things that will really change the playing field for synthetic biology, both inside, directly related to the science and the tech, and outside. And the very broad theme that came out across all the breakout groups was what really matters is the very first thing that is tangibly produced by synthetic biology and how it's viewed by the many publics. So if it is something that is very instrumentally beneficial to human health and it's widely available, and there's a tremendous sense that this is a great good for humanity, then synthetic biology will have a very different public reception than if it's say, a very trivial or cosmetic application, or something that it maybe comes from the military instead of a military application, instead of a human health-oriented application. If there's an accident or there's some kind of unintended consequence, negative unintended consequence, the game would change completely in terms of the public's appreciation for the risk-benefit ratio with respect to synthetic biology. And those are things that come from lessons learned in the past as well. So that was a really clear theme that went all the way through these discussions. Another one was that if there is an application that's beneficial, rather than a negative consequence, it will really matter how those benefits are distributed. So if the public sees that they're not widely distributed, or they're distributed in a way that stigmatizes a particular group, or they're captured by intellectual property and private ownership, and so that there are benefits that are seen to accrue to only a very small segment, that will be also a game changer in how the public views the benefit-risk weighing ratio of synthetic biology. So that was the first breakout session. The second breakout session, the one that actually broke down according to sort of academic or professional competencies, the first group, the legal and economic group, there was really a lot of very rich information that came out of this group. One of the major themes that's run through this has been an intellectual property theme that you'll see. There was a lot of concern about how intellectual property was going to be applied to synthetic biology. And the background to that is that synthetic biology is really based on information. So when you have genetic sequence, you're able to create things using synthetic biology. And so the information then that's available to you sort of breaks down geographical barriers in areas like where there have been concerns about bioprospecting and biopiracy. So if you have the sequence information for some kind of natural good or organism that is in a country where it's heavily protected and considered indigenous, and you reproduce it outside the country, are you engaging in biopiracy in a way that hasn't been thought of before, rather than going in and collecting it and bringing it out and owning it? So it kind of changes the background to the concept of biodiversity and biopiracy issues. And intellectual property, synthetic biology, if you see it as a thing and you're going to patent or control through intellectual property various pieces of it, then it really loses the appreciation that synthetic biology allows you to have a toolkit, like a platform for other kinds of applications. So how the intellectual property system will deal with synthetic biology was a really open question for the legal and economic group, as well as the regulatory structure under which synthetic biology might be regulated, which I'll talk about a little bit later in the synthesis. That was another big issue, just a question right around what kind of regulatory structure. The second group looked at societal and public policy issues, and here they underlined a number of things. Foremost among them was the need for open and objective information about the underlying science. So rather than, as the science is progressing, rather than having the applications, the really beneficial applications, drive the communication with the public, it's the actual underlying science, what's actually happening, why we're doing it, what's working, what we still have to work on, including the unknowns and the things that we're not there yet on. We're working on them, but we haven't figured out how to do it yet. And John Glass, in his plenary session, his introductory plenary session, he's a scientist with the Venter Institute, did an excellent job of that. He was able to tell us, we started working on this microbe and it just didn't work. And this is why it didn't work and the mistake we made. So we had to switch over to something different and we weren't sure we'd get the results we wanted, and these are the difficulties we had. But it was very transparent information about the science, which is really meaningful to people who are non-scientists but who are interested in what's actually happening. So they underlined that need for objective information about the science itself. And input from a wide range of communities, not simply a smaller group of people in the know, but a wide range of communities, including religious and environmental communities. And they also suggested that initiatives like the DOE and the Sloan Foundation were able to do by bringing together researchers across different disciplines, but initiatives that allow cross-disciplinary work to be exposed to each other and to bridge the gaps, not only between those people working in the field, but between scientists and journalists and people that are interested in synthetic biology, whether they're working in it or not. Those kinds of initiatives would be invaluable in being able to understand and address societal issues in a meaningful way. And then the third breakout session was the Public Understanding and Engagement Group. And one of the big themes that comes up, which I'll talk about in a minute, is the idea of what synthetic biology actually is and what it is not. And so they started talking about this, that there needs to be some common understanding of what synthetic biology actually is and how it differs maybe from even things like synthetic genomics. Is that a subset of synthetic biology? Because they're both used. And it's really difficult to talk and engage with the public unless you start with a common understanding. And that is significantly more difficult than it appears to be when we're talking about synthetic biology, which I'll talk about in a second. Also, this open discussion of potential benefits and risks no longer really is sufficient to talk about the benefits and not talk about the risks, or talk about the risks and not talk about the benefits that are foregone if you don't move in a certain direction. So that kind of open discussion about benefits and risks and unknowns needs to take place. We have a very much more sophisticated public than we might have had, say, 30 years ago, very well aware that there's always unknowns and two sides to the coin, et cetera. So that kind of openness is absolutely critical. And that paternalistic approaches, and by this I mean the approach where you say, if you only knew what I knew, you would agree with me, which is how we might want to bring up our children sometimes, but doesn't work very well with this kind of engagement. So that kind of paternalistic, I have the information, I'll share it with you and you'll get on board, not only is not meaningful, obviously, but it can turn toxic very quickly. And I think we've experienced that in other biotechnology applications, so that everybody was well aware that that simply isn't the road forward to meaningful communication. Now, the meeting synthesis was something that I kind of traveled around during the meeting, I think with Dave as well, and tried to sort of hit all of the breakout sessions and to synthesize what we'd heard in the plenaries and all the Q&A and the discussion, and there was a very rich discussion. And the following themes were sort of the highlight themes. There were many things covered, but the definitional issues were absolutely key and came up over and over and over again. What was fascinating was one a synthetic biologist saying to another, well, I'm not sure that what you're doing is synthetic biology. I think what you're doing is like metabolic engineering. And he'd say, no, I'm doing synthetic biology, but I'm not sure what you're doing is synthetic biology. So, and so there's really, it's not just that there's not a common understanding in the various publics, there's not a common agreed definition of what falls in or what falls out of synthetic biology in that community on its own, not even commonly agreed on. So, and in particular, do you, like if you just self-identify, I'm a synthetic biologist, does that make what you're doing synthetic biology? What about the DIY community and the iGEM students? Are they members of the synthetic biology community? How do we define that? They clearly are members of some community doing something that they think is synthetic biology. Does there have to be some kind of professional standard, or is it just enough to say that you're doing it? So that is a threshold issue that just hasn't been covered yet. I don't think we have any kind of clear answer to that at this point. And then this public engagement communication, since we're talking about societal issues, over and over again it came up that there are, of course, multiple publics. There's not one homogeneous public, that there are multiple publics, and to our detriment, we exclude groups like religious groups who many of whom may be very much in favor of what they understand to be synthetic biology, but many of them may be very against it, and sort of marginalizing any one group and not inviting them to the table doesn't enhance any aspect of public engagement at all. We think we've discovered that in the past as well. And that it's also exceedingly meaningful to have the challenges that are posed, or the unknowns, or the risks, articulated by the people who are actually doing the science. So, because what that does is it really shows that the scientists themselves are concerned about what they're doing, and curious about what they're doing, and motivated by the right reasons. They want to do good science that brings about benefits to the world writ large. So that involvement of the challenges and the risks articulated by the practitioners was something that came up over and over again as well. And then this concept of game changers, what might look like a game changer in synthetic biology? Well, we had a number of things that were mentioned, repeated numerous times, including new fuels, which is a more obvious one, but also applications to the human microbiome. So synthesizing some of the microbes that are part of our human system, and how that might improve human health, how that might change certain aspects of human health or disease management, et cetera. And that was actually really an exciting part of the conversation. And I think a number of people mentioned over and over how that would be a real game changer if that was where the application space for synthetic biology ultimately moved. And of course, environmental release, hugely important. Tackling the issue of environmental release, understanding it, or at least understanding what we don't understand. Attendant with that as well, first success, first failure, clearly, as I mentioned before, game changers. Then this issue of regulation that I flagged earlier kept coming up, and the question of how it should be regulated. Does it fit into our existing coordinated framework? Does it need its own framework? Is it something that, rather than being a single application, it's a platform, it's a toolkit, et cetera? Is it something that can be centrally regulated? Is there some way that we can actually include discussions about welfare or well-being as part of that regulatory process if we were creating a new one? Do they always have to be sidelined outside the process? So this kind of concept of how we could probably possibly design a regulatory system that would be dynamic enough to actually provide meaningful regulation to an ever-changing scientific field. And I think that's a question that's been raised before, but it was a very pressing, interesting question to the people around the table at the workshop, including the need to study other regulatory approaches used in other parts of the world, and other IP approaches like open source, et cetera, that might be better suited to the goals and being able to realize the benefits of synthetic biology as well. So that the need for more research in that area. And then finally, how ELC research can be meaningfully used in this context. So it's been around for a while now, ELC research. Should it be embedded? Should it be something that's part of all the work? Should it be something that happens at the same time or afterwards as a ex post facto analysis? And how do you actually find measurable contributions from ELC research? That's been a really difficult question. How do you understand whether it's making a difference in any real sense, ELC research? And then how communication can be fostered about the issues raised of ELC. And then finally, before I turn it over to Dave, what I mentioned before was that we did this gap analysis, and the question that we asked all of our participants was, what important issues or questions do you think still need to be addressed in the field of synthetic biology as they relate to societal issues. And we created 4 divisions, just the divisions that seem to map out on what we've been doing throughout the last 2 days: the legal economic, the IP related, the public policy, and the public engagement and communication. And we asked everybody to take a few Post-its and write down the things that just hadn't been addressed or they felt really were much more urgent now that they've been through the workshop, and to post them. And I think Dave's going to talk about those.
Great, thanks, Laurie. So what I'm going to do is actually let you see some of the Post-it notes. So yeah, there's no attempt here to scrub these for good grammar or anything, but I think it'll give you a sense of kind of the things that people were putting up on the charts near the end of the second day. So the legal economics. I think the other thing, the exercise interesting to do would be to think back to some of the meetings I know some of you were at, we did on nanotech over the years, and ask the question, were these the same things that came up then? Or are these things that are actually just kind of unique to synthetic biology? To what extent are they generic to new technologies? So this idea of anticipatory adaptive governance, I don't know how many workshops I've been to on that topic in the past 5 years. I think we still haven't figured out what that is, how to do it. But this issue of pacing governance to fast-moving technologies, this issue of how do you even incorporate these kinds of ethical sort of concerns into governance and regulatory structures. To what extent, what does government involvement look like, actually? We kind of know what that is in an institutional sense, but people started to raise that in terms of the do-it-yourself biology community, which is essentially a community that exists outside of government institutions. Somebody wrote this. When you actually look at the publication, there's copies of what people wrote in. So we got some kind of interesting, this is what came in on one of the challenges somebody ran into. I was teaching a course at a law school a couple of years ago. A student came up to me in the break and said that he and his buddy, a master's bio student, had bought a bunch of used equipment and were trying to create and design some new critters in their garage. He was asking me about regulatory issues. I was bothered by how cavalier he was and how haphazard they seemed to be, but I ended up saying nothing. So it's interesting that this is obviously a fairly large challenge, people are running into it, but it's not an area where, in sort of in classic law sense, there's a lot of answers because we've moved outside of the normal institutional structures of universities and scientific laboratories. What does progress mean? That came up a lot, but again, that's something I think that we met in nanotech and cognitive neuroscience and IT. So those are some of the stickies that were up there. IP issues, are there particular ones related to synthetic bio? What IP regimes might work for disruptive technologies? There's this kind of tension between sort of the biotech approach to IP, which is involved usually control of IP, to open source movements like BioBricks. And I think that is something that's relatively new. We saw that in the IT sector, but it's reappearing again here with synthetic biology. How does the current IP regime affect synthetic biology research in the way that it promotes innovation or constrains it? And how can these historical conflicts between, say, the classic life science approach, which was let's grab as much IP as we can, to the engineer's approach and the computer scientist approach, can they be resolved, and how would that look like? So again, the IP issues were, I think, very prominent. They were raised again and again. Public policy. Should we label? Right? I mean, that's a classic one. We heard it with nanotech. Do we label the products of synthetic biology? How do we ensure that the intended effects are achieved and unintended consequences are benign or limited? I mean, that's a classic thing we've struggled with for 30 years in technology assessment. How do we educate decision makers? Again, that I think is a fairly common one. And I think how can we come up with a cleaner, this is this issue of definition, whether it's between the scientists themselves or between scientists and the public. The last sort of bin that we filled up was public engagement and communication. Can you make a case for synthetic biology ELC work? Why does it matter? Why should anybody fund it? How would we engage the public? What kind of interesting ways might we be able to do that? Could we better engage on risk assessment and integrate public views into risk assessment or hazard identification? And with regard to synthetic biology, what should be communicated and by whom? So again, one of the early comments that came in when we did the pre-questionnaire was from someone who had just started working on SynBio. I'm constantly challenged by people's reaction to my describing my job. My friends want to know if I'm engaged in genetic engineering. Genetic engineering's got a bad name. Synthetic biology is also a loaded term and a terrible label for our field. Worse, nothing could imagine what a worse name could be when we started talking about this. If we can't converse with the public about what we do, we face grave obstacles to working towards positive solutions. So these were, again, some of the kind of stickies that got thrown up on the wall. So we were thinking about kind of how do we keep this process going, because it's not complete. I mean, this was a 1.5-day workshop. There were a lot of people that really weren't there. We didn't have civil society groups. We didn't have any, I think, religious groups. There weren't enough representation, I think, from industry, from the financial sector. There weren't enough people internationally. The science is international now. A lot of the debates will be international. So I thought what we're going to try to do now is kind of actually continue the sticky exercise. And when you go into the website now, what we've done is actually launched a polling or survey instrument that we're going to keep up for about a month now, which essentially will allow you to add to this list of what you think might be, really important emerging issues. So the goal is, again, you can actually create a new bin. We've got these existing bins, the legal and economic bin. We've got a bin for policy-related outcomes. We've got one on public engagement. If you want to create a new bin, we haven't thought about creating a new bin. But I think what we'd like to do over the next month or two is actually just open this up. We're going to send out a mass email to our synthetic biology list, and we would encourage anybody that gets this that you resend it, retweet it, whatever you want to do, paste it on, spam people with it. But the idea is actually to sort of get a lot more input into this process, because there's just no way you can do this in a day and a half, no matter how well you try to do it, how well you facilitate the process. What we're hoping to do maybe after a month and a half is look at what's come in and kind of consolidate, get rid of some of the redundancies, and we'll repost with prioritization so that people can actually vote. So we think it's actually very important to prioritize these. If I'm a funding agency or if I'm a foundation, there's never going to be enough money for me to fund all this. So there's a lot of players in the federal government. Obviously, you've got the DOE. NSF has got large investments in synthetic biology. DARPA just started a new program called Living Foundries, multi-million-dollar program. So there's agency money out there. There's foundation money. But again, it's going to be relatively limited. So if somebody is going to be sort of asking the question, gee, I'd like to engage in the societal issues, legal issues, what would I actually fund? We're hoping that this kind of process, if we run it for at least a month or two, might be helpful. The other thing we'll be asking, to the extent people want to give it to us, is some information about who you are, so that we can actually take a look and see what are some of the top issues for people in government versus NGO folks versus industry folks, and we can kind of statistically sort these. I think that's going to be, fairly interesting exercise. So we obviously need your help with this. You can, if you go into the website on the event, there's a click on button, and you'll go immediately to this survey instrument. You start filling it out and send it back. We will send out to our email list on synthetic biology, and we'll also, I think, hit some of the nano folks and S&T folks. And we'd love to have your help in terms of just propagating this out there. Again, in a month or so, we'll close it down and put up a poll where you can actually begin to vote. And then hopefully we'll have some information we can share with you in a 2-month period or so. And that information obviously will hopefully be useful for agencies thinking about funding into this LC area or foundations. I think that's, the online survey is that actually are the event page on our website. But most of you, if you're not on our email list, just leave us your email. Tomorrow or the next day, we're going to shoot out an email to let everyone know that this is up and give you just a click-on link so you can go in and start filling out, give us ideas. So that's it. I think we'll take we've got almost a half an hour for Q&A. So we'll get you microphones. Just let us know who you are and where you're from.
I'm Mark Segal from EPA, and I was interested to see that there were two elements of interest to me. One, nomenclature, and the other, regulatory oversight. What I was concerned about or wondered about is whether or not those two were ever tied together during the workshop. And the reason I say that is that 25 years ago, in the development of our biotechnology regulations, we used the term chemically synthesized with respect to genes as part of our definitional elements. So that gives us a handle on now being able to deal with that set of synthetic biology involving chemically synthesized genes for certain kinds of products under certain circumstances. The rest of the federal government may or may not have those kinds of terms. So I just wondered if there was a mechanism, or I wondered if people in the workshop put those two items together to recognize the influence of the definition of synthetic biology on the way we might be able to regulate.
My recollection is that that particular discussion did not happen in the group, that there wasn't a delving deeper into the coordinated framework to see how you might either bring it into the framework using language or take it out. I don't recall any of that taking place. And there wasn't a tremendous discussion about how it fell into the coordinated framework either. The discussion was at a little bit higher level on that particular issue, talking about would it be better for there to be something new rather than to have it fall under the coordinated framework. Those are certainly my recollections. Do you have a different?
Yeah, I don't think people necessarily there was a lot of discussion, there's a lot of disagreement about what it was, synthetic biology. So we are sort of at that level of, can we actually agree on the definitions? So the nomenclature is, it plays a key role. We went through this in nanotechnology, We define it as something, change of behavior under 100 nanometers. 100, what does that mean? Along what axis? Is it an aggregate? All of those things have a huge impact on whether EPA or anyone else regulates, whether they see it as a new substance or falling in, sort of generally recognized it's safe. So I don't think, that's an interesting area to drill down in, because I think it's going to be actually pretty critical as we start thinking about regulating these products, is how will the EPA or any other agency actually define them? So that would be an interesting thing to follow up on, to just have a bunch of people sort of talking about that, I think, especially with the scientists who, as at this point, haven't really reached agreement about what is synthetic biology.
Thank you. Sally McCammon from USDA APHIS, and I'm also from a regulatory agency. Just to follow up a little bit, two questions, one a question and one a comment. How does DOE define synthetic biology? Does it define it as when they're requesting grant proposals? Is it metabolic engineering or is it biobricks? So that's there must be some realm of projects that they're looking for. And two, as regulators, you hit it on the nail on the head. We're looking primarily at products. So the big debate back in the 80s was, well, do we regulate based on the process? And if we do that, how do we identify risks, which sounds like what the conundrum is here? Or do we look at the particular products, and are there any new risks that are being proposed, or is it just a huge amount of uncertainty that people are uncomfortable with? From my perspective, the USDA system was put in place to kind of give some comfort that uncertainty was being looked at by the objective government representative of the public, to just verify that what the scientists said that they were doing to plants or microbes, it was what they were doing. So I think the definitional issues can be a little bit of a double-edged sword if you're just talking about a bunch of tools, or are you talking about particular products?
I think one of the things that came out as well in our discussion about the definitions was that this is really, it's not sort of a clean category. It's something that builds on other things and other knowledge. It's sometimes doing something you've done before but using a different process. So and having the similar outcome. So as you said, it's really not something that sits on its own off on a shelf. It's built on other things that have come before, and that's why there is, in fact, a lot of overlapping. And the overlapping is healthy for the science to develop and to become, interesting in a number of ways. So the definitional issues are there for a reason.
Yeah, next week we're actually having a workshop where we're taking a look at a synthetic. It's a cyanobacteria that's being developed at Harvard that could be used in if it's scaled up, probably in mass sort of open ponds, and it'll produce sort of sugar-based products, inputs into potentially the food system, chemical system. And we're going to be bringing in probably 6 or 7 ecologists just to talk through and think through issues like gene flow and what happens if this got out, or can it be contained, to talk to the scientists. And there'll also be a number of people from the regulatory agencies there and the NGO community. So we're trying to do a number of these exercises where we're actually trying to go upstream in the scientific process before a product ends up essentially at your doorstep, and dig also a little bit deeper into the production process, which we haven't necessarily regulated in the past. But I think if we look at what's raising a lot of issues, this issue of potential environmental release is a huge one. And so the idea is actually to put a lot more sort of intellectual horsepower in terms of looking at what are the potential downsides of uncontained use of this. And you probably will have to have uncontained use if you're really going to scale it up to the point where you're making biofuels or enormous amounts of high value chemicals. So that's something we're very interested in doing, is sort of going and taking a look at some of these, unpacking this question of what about the process, because the process is changing. JD?
Yeah, J.D. Hansen from the International Center for Technology Assessment. I've already talked with you about civil society, so I won't ask questions about that, but I will ask about the workshop that was being reported on. I'm curious about why neither the Department of Energy LC office or the NIH LC office were in the conversation. They've actually got a lot of experience and spent an awful lot of money on the Human Genome Project, and I think it would be useful for this project to get their observations. And when I asked the DOE ELCs person if she knew what was going on with synthetic biology, she says, "We're doing what?"
So I don't really have an answer to your question of why they are or not involved. I know that the DOE ELC people that we know were the people who suggested that we actually put together this workshop. So they were integrally involved in fact, in spearheading this with the Alfred P. Sloan Foundation, and without them, it wouldn't have happened. So I don't know how to answer that specific part of the question, but I know that there is lots of great work that's being done. We didn't have everybody by any means around the table. We were really well aware of that at the time, and that's of course a shortcoming that we would like to try and get more voices involved, and we'd like to try and address that by getting more voices involved. But anybody that you know or that you've met that you think would be interested, including people who should know what's happening, by all means, JD, loop them in.
I would also urge, I've had some very interesting conversations with recently retired government people. And I think some of those that have actually been involved in trying to implement the coordinated framework that are recently retired may be more forthcoming about its deficiencies than folks that have to try to make it work now.
Sure, yeah, absolutely.
Yeah, and we have a few of those coming next week to this workshop. I'm a big believer in retired government folks in terms of opening up the debate. Yeah, Peter?
Tim Stevens, National Sheriff's Association. I guess my broad question is, were there any things that surprised you And then just a comment to maybe provoke you a little. My quick read on this, there isn't a lot of urgency described in what the research needs are, other than researching more policy positions around synthetic biology, rather than what we may actually translate into real products that would benefit society. So that's a comment, and I just don't know if you got some thoughts about what was surprising in the workshop and what is to be built on.
Well, so the first thought that comes to me is that I, having been sort of working in this end of synthetic biology for a while now, there's a tremendous sense of urgency from the synthetic biologist's perspective about the intellectual property system and the regulatory system. But the intellectual property system trumps even just the overall regulatory questions. So while that urgency isn't translated here in our discussions, that is the theme that they keep coming back to over and over again, that it's not equipped to deal with what we're doing. It could shut down our ability to use these platform tools if it's applied in the way it's been applied in other venues and other areas of life sciences. We need something sui generis. And then those of us who work in IP recognize some of these anxieties from other areas of science. It's not new in the sense that that discussion about the IP system being broken or not being able to adapt or not being appropriate isn't itself a new discussion, but it feels very new to the synthetic biologists. And there's a real sense that if it's applied, the first application of the IP system in a negative way to close some doors could ultimately have significant long-term effects on their ability to use the tool to its full extent, to actually realize the benefits, the power of it across a broad breadth of biological platforms. So their sense of urgency seems to be in the IP area. I would also say that this, although it's a recurring theme with other types of biotechnologies, this sense that we will never be able to know everything about how the applications will manifest in the environment, and we won't even be able to know what all the risks are. We haven't gotten, there is no answer to that yet. There isn't an answer, and so it keeps coming up over and over again. What do we do about the fact that we know that we don't know, and how do we communicate that meaningfully to the public, and yet help the public see what we as synthetic biologists, I'm speaking in their shoes, as the tremendous benefits that would be foregone if we don't actually accept some risks and the fact that there are some unknowns. So they still are very much struggling with that over and over again. And that's not new, but it is sort of they recognize that it's not something that they can sweep into the corner, that it has to be out in the front of the discussion. Do you have an answer as well?
I mean, I think the IP issues are front-loaded. I mean, that's the one thing that I think in terms of urgency. But the one behind that is definitely this communication issue, that people just don't have a sense of how do we talk about this, and how do we talk about this in a way that is not going to scare people or alienate people. And so, how does that tie into research? I mean, I think certainly, I think. The late 80s, I had an office at EPA, and the office next to mine was the Office of Risk Communication. And that office is gone, I think, unless they hid it somewhere. I can't find it. But I remember EPA actually making pretty significant investments in how to sort of talk about, communicate risk issues. And they funded a lot of seminal research that was done back at the late 80s and early 90s. By people at Carnegie Mellon and other universities. So I think that in terms of research needs, I think some of this falls in what I would call the social science basket, and that's a basket that's being pretty eviscerated right now. If you look at the areas that are under attack by an NSF right now, it's the social science and economics and behavioral directorate. So I think one of the things that's lurking in the background of this is kind of some of the answers might come out of sort of areas like such as social science research that we haven't spent a lot of time investing in. and some of the stuff that's related to the IP issues. So I don't know if we've gotten any EPA folks, maybe you can tell me where that office has gone, but I don't know.
The office is gone. Derry's still plugging away at a lot of the stuff.
You know, the other issue that came up in concert with the issues that Dave was just mentioning is the issue of regulation from the funding end. And so there was this question that came up several times of, you know, should there not be a more sort of competitive position of the United States, that the government is taking a bigger chunk of the funding activity in this area? And if so, how would they choose how to prioritize, how to fund, how to choose them, and then what kind of IP would be appropriate, or what kind of strings attached to the funding so that this was in fact broadly available. And also not a brand new theme, but something that was very much part of all the discussions about regulation and IP.
Yeah, Peter?
Thank you. Peter Utro, Environmental Protection Agency. I was thinking back to the 1980s, mid-1980s, about the same time in the biotechnology area as we are probably in now in the synthetic biology area. And I was thinking about the relationships between national political approaches to the issue and what evolved into the dispute, or, well, I guess it's a dispute, between those who adopted a precautionary principle approach and a risk-based approach to potential regulation. Are you starting to see any evolution of national trends in one direction or another, or any kind of a reemergence of this as a politicized debate rather than at this point just one based on genuine concern and inquiry?
You know, so the one aspect that I mentioned really briefly that comes to mind is that there's often been the distinction made between how Europe approaches risk and how the United States approaches risk, and that the discussions that and the burden of proof that's required, whether it's, you know, a preponderance of evidence or sound scientific proof, so that the proof thresholds are different as well, and the focus on product versus process. But one of the things that I think people now recognize, at least in our discussions, it's come up several times, is that having a regulatory system that is designed and for other things and is now used as a coordinated framework for biotechnology, and that doesn't allow for broader-based discussions about non-harm-based risks. So it doesn't allow for these broader issues of well-being or even things like animal welfare. So it doesn't have room for those broader discussions, puts a significant amount of pressure on a new type of biotechnologies to have those discussions in a different forum and to be seen to be having them legitimately. So if they're taken outside of the regulatory process, which has a necessary legitimacy because it's part of the government regulatory process, that seems to sideline them and make them of lesser importance. And so as long as you talk about them, then you've done that, you've ticked that box, and you can move on. And I think there's a much greater understanding now that in some ways it would in fact be helpful to have those discussions raised to a level where they weren't always a sideline to the regulatory process. At the same time, you don't want to have them take the place of sound science. So finding that balance where you still have sound science as the backdrop of your regulatory process, but also allowing those parts of those concerns to be part of the process and sometimes outweigh the process. How you put those together into the regulatory structure is still a really open question. If you don't put them in the regulatory process and they're overwhelmingly part of a public veto, you'll find out later on in the process after the money's been spent and the regulatory hoops have been jumped through anyway. So to your detriment, unfortunately, you have a system sometimes that doesn't allow for those things to be voiced in a legitimate way.
I think the other thing that plays in, which we've discussed before, is just the different patent systems. So the European Patent Office is integrated part of the French law that allows them to think about the potential threats to the public order of a new patent. Public order is defined fairly broadly, but it's something that the U.S. patent system is essentially kind of put outside almost completely. We don't really have a discussion in the patent system whether there are particular kinds of social issues or deep religious or ethical issues that might be brought up through patenting and innovation. And that's another difference between the European and the U.S. system. It's just the willingness to sort of open up the discussion to broader social issues. So I think those things will play in, as well as the just potential general cultural pushback that's related to uneasiness and nervousness about anything genetically modified in Europe that goes kind of beyond just the political regulatory system. Yep.
Dave Rabinowitz, and I'm wondering, I have two quick questions. First, I'm wondering if looking at synthetic biology versus genetic engineering, nanotechnology, or even robotics and artificial intelligence is a distinction without a difference from the point of view of ELC. And if defining it is really that meaningful a task, number one. And second question, has anybody proposed any concepts at all for regulating non-publicly funded research in synthetic biology? I find it hard to believe that that's even possible.
Well, yeah, one of the debates we had here, I think about a year ago, when we released a report on ethics and synthetic biology, is do we need bioethics, neuroethics, infoethics, nanoethics? Is there really a distinction? And I think at that point in time, the ethicists were hard-pressed to actually figure out, is this raising any new ethical questions? Except maybe, this issue of could we create a totally synthetic life form? What does that actually mean? I don't know if you've got any. There seems to it's a good question, and it's a question that actually people are interested in debating, whether we need a new form of ethics every time we have a new technology.
And I think that there are actually projects that are ongoing looking specifically at that, like are there any new, or is there something new here? And it seems to be that it really depends on what the application is. And it may be that the application itself, if you are thinking really broadly, you can think of something that, so they talk about, the name of it escapes me now, but they talk about creating life forms based on a totally different structural DNA, with 3 base pairs, and I can't remember what the name of that is, but there's a name for that. And so where you create life forms that are actually on a completely different platform, that might be something that raises, and how would they fit into our society, et cetera? What would you use them for? Does it matter if you can control them, et cetera, et cetera? So where you can think about these kind of really different kind of application spaces, then maybe, yes, there are really new issues raised. I guess it seems to be that if you get stuck on the issue of newness and you constantly come back to analogy, well, we've been there before, we've been there before, we've been there before, ultimately is an unsatisfactory answer to the person who's really concerned about this first and foremost. They don't really care about the discussions that you had before and that this is similar to it, but they really care about you answering how this impacts them now. So I take your point completely, because we've spent a lot of time talking about this question of, is there something new here? But I think ultimately the work still has to be done to answer maybe the same questions again in different application spaces. And then you had a second question. I'm sorry, I don't remember. And could you restate that question? Oh, regulating private enterprise, right? So there is a backdrop, obviously, of biosecurity regulations as well that might have some implications for what you're talking about in the private. Are you talking about the garage hackers, the sort of the DIY community, or?
Synthetic drugs are being created all the time.
Oh, okay. So I think the answer to some of that is you look at the systems that we already have in place, and a lot of them will fit into the regulatory systems that we have by analogy, like they've done in the coordinated framework. And so I know with the folks at APHIS and at EPA that we have in other areas ongoing discussions with them about things that we want to do, and they constantly are working by analogy. We've just written this letter, and it's about, Kentucky bluegrass or whatever, and so you'll find that we're moving in this direction with this, or so I do know that a lot of new things do fall under the language, as you were saying, Mark, that the language captures a lot under the already existing regulatory systems. I'm sure there are things that fall out, and there are certainly groups of people that fall out that don't see themselves as covered, which is probably more important than whether they are covered or not. They don't even know they're covered or see themselves as covered, and that, I think, is perhaps a bigger problem.
Take one more question. Yeah, way in the back.
Alona Baki, AAAS fellow at TPA. So I'm wondering, I was delighted, Dave, as an ecologist, to hear that you have the group of ecologists involved in this experiment you were describing for the ponds next week or something. But given that environmental release was one of the greatest concerns expressed, and that the implications of environmental release are really different depending on the definition. So if we're talking about chemically modified, genetically engineered, or de novo synthesized, are there any efforts to create a work group of ecologists that are creating a more generalized taxonomy and looking at the various threat levels, kind of maybe predicting and looking at them related to the different possibilities and advancements or definitions?
Well, what we're sort of testing is the EPA has developed something called a comprehensive environmental assessment framework, which they've been trying to test on nanotech. But it's a way of actually getting people to look across the entire life cycle and then sort of drill down into particular areas where there might be risk being created. And so part of this exercise is actually to try to test that and see, is a decision-based tool, could we actually get some of the it would be useful for the scientists to be able to kind of sort out and help them think about what they're doing.
Right,
both across the life cycle and sort of doing a deeper dive. How many decision rules would it take to avoid potentially some issues? Because unfortunately, we'll never turn all of the synthetic biologists into ecologists. So can you come up with a frugal decision tree that'll allow them to sort of sort out, well, or at least think early about, well, gee, if I did that, maybe I should be thinking about that in terms of ecological impact. So the idea is to try to, and the other thing we're trying to do is if we can compress and create a decision tree that actually will help us mitigate risk, can we then put it in, for instance, into the iGEM process with the students? Because as the students actually go through that competition, they're required to do a kind of analysis of the environmental impacts of their whatever they're creating. But the tool right now is relatively weak. So can we come up with a tool that's much more powerful, that's been peer-reviewed, and get that into a fairly simplified form? We obviously have done this fairly well in some clinical settings where we figure out, there's 4 or 5 decision rules that doctors can apply that can save a lot of lives, and they don't actually have to drill down do an entire 20-step fault tree analysis. So we're trying to figure out what are some of the key questions that the scientists would have to ask if they're creating some sort of new critter or it might be released. They would say, I better go, if I don't have the answer, who should I talk to about this? Because it's raising some issues. And can we do the same thing in a simpler form for students who will graduate and then become the next sort of generation of synthetic biologists? So that's the exercise we're doing. We're taking a tool that's actually been used by the EPA fairly extensively, or at least thought through in terms of the steps. And can we take that and kind of see whether it applies? If not, where do we change it? How do we tweak it? Can we reduce it, simplify it, that sort of thing? And get it embedded into the research process rather than having the downstream people come in really late and say, something's wrong here. You're going to have to change your process. I can't do that. I've already invested $100 million. Right. That technique usually doesn't work. So we're at 1:30, 1:35. Thank you. And thank all of you for trudging over here through the heat. And please, we'll send you, if we have your email, you'll get an email from us the next few days to kind of contribute to this list we're trying to create. Feel free to send the link on to other people. And hopefully within a month or two, we'll be able to share some results with you in terms of how we've been able to expand the list, and then we're going to actually try to engage you to prioritize and contract the list. So thanks again.